Specifically, the one which shocked me to the core was like, if you notice over here, there are like almost 1 million plastic bottles are purchased every minute. So which takes almost 100 years for it to degrade. And that is concerning because we do not have that kind of a resource to spend on remediating plastics from the ecosystem. So this helped us to focus on a pollutant which we were planning to work on, and we chosen microplastics as a target group of pollutants, in which specifically we chosen dibutyl phthalates. Dibutyl phthalate is not an end product, but basically it's a compound which is used to make flexible plastics, like polyvinyl wrappers, plastic bottles like water bottles, or PVC pipes, resins, whatever that are like flexible. Dibutyl phthalate is actually a flexible plastic reagent, so which is almost very common than the specific plastics like HDPE or LDPE plastics, which are for its intended for specific use, while dibutyl phthalate is more common than we could imagine. So these plastics, they do tend to get accumulate in brine, because that was also like crossing blood-brain barrier was one of the shocking things, where the traces of microplastics were found in human brain, lungs, spleen, stomach, kidneys, skin. Even our skin tend to absorb into microplastics. And because of these accumulation of microplastics, specifically dibutyl phthalates, there are several harmful impacts. So specifically neurodevelopmental disorders, hormonal and thyroid functions, cardiovascular diseases, respiratory diseases like asthma, and male and female reproductive issues, and carcinogen, which is a very common carcinogen, like it's one of the common carcinogens, which raised to the top 10 level of pollutant of emerging concern. So definitely the health impacts are far more serious than the advantages which it provides. So the removal methods, which are like either from primary sources or secondary sources, it can be broadly divided into 3 categories, like physical, chemical, and biologicals. But each of these methods, they do have their challenges, like they either slow and time consuming, or either like exorbitantly expensive, or they do produce secondary toxic intermediates, which leads to other pollutants. And also it is not fully understandable to how to tackle these secondary intermediates, which are more toxic sometimes, and how to face those issues in the ecosystem. It's not yet fully evaluated. So one of the method which we propose is like bioelectrochemical systems, which is basically like a hybrid system of combining biodegradations and electrodegradation. Basically, we use electrode stimulating bacteria to have an enhanced degradation. So in this work, we first isolated the bacteria by collecting leachates from the landfill sites. And with the landfills, the specifics of the landfills were majorly composed of plastics, heavy metals. So we thought that could have a potential bacteria that could be used for microplastic degradation. So once after it was isolated and identified using 16S sequencing, we went for degradation optimization by varying parameters such as pollutant concentration, initial pH, temperature, inoculum dose, and applied potential. While this experiment were going on parallel, we also evaluated the enzyme quantifications on the key enzymes that play a major role in degradation of plastics. And to understand the mechanism, this helped us in a way. Followed by these steps, we wanted to elucidate the pathway, which was one of the important steps to understand, because as I said earlier, the production of secondary toxic intermediates could possess a serious threat. So we want to elucidate the pathway on which the parent compound of dibutyl phthalate is broken down into simpler fragments or not. Followed by the treated effluents were checked for their toxicity assessment across multiple models like in silico, cytotoxicity, and artemia, and mice as an animal in vivo experiments were done. So this was the methodology overall schematic representation of the methodology which we employed. And as of results, the screening was done. In order to isolate the potential bacteria capable of degrading dibutyl phthalates, we were supposed to isolate a bacteria that should be having a resistance to dibutyl phthalate. So we used dibutyl phthalate in minimal salt media in absence of any carbon source, and utilizing dibutyl phthalate as a sole carbon source. These bacteria were isolated, like randomly 20 bacteria which were isolated, screened and selected, of which LLS04, which was capable of utilizing dibutyl phthalate to the maximum compared to the other strains which were able to grow. And later we identified this bacteria to be Bacillus subtilis. And this LLS4 dibutyl was utilizing carbon source, which we increased the pollutant concentration in order to evaluate its minimum inhibitory concentration, like to have a rough idea of the range of pollutant degradation which we could do. And as you can see, like there was a decline in bacterial growth at 48 hours with DBP as a sole carbon source. Still, until 500 milligram per liter, there was substantial growth was seen in the utilization of DBP as a sole carbon source. So we had a rough idea of what range we could work on it. So further we proceeded with biodegradation, electrodegradation and bioelectrodegradation, like the two separate methods compared to the combination of hybrid methods, how their degradation efficiencies were changing due to the several other factors. From this data, you could see like electrodegradation was not much of a removal method strategy because they are very slow in terms of degrading dibutyl phthalate, while biodegradation was one of the strong candidates as a bare minimum, but the problem was they were not able to degrade more than 85 to 90%, while dibutyl phthalate was almost completely removed up to 99% in bioelectrodegradation at day 8. So when we try to study the kinetics plot, we could see that the rate constants of biodegradation and electrodegradations were less than bioelectrodegradation. In fact, the linear sum of biodegradation and electrodegradation were less compared to bioelectrodegradation combined. So there is a significant synergistic effect, which is always helpful in removal of pollutants, especially in terms of pollutants that has to be removed in the ecosystem at a short span to maintain cost and efficiency. So thus, comparing with these three methodologies, we tend to proceed all further experiments with bioelectrodegradation. And the reason for why bioelectrodegradation was much better than biodegradation could be explained based on the two key enzymes like esterase and dihydrogenase. These are the two key enzymes, not specifically for the removal of dibutyl phthalate, but they are the enzymes which are usually used in removal of any complex organic compounds that easily helps to break down complex organic carbon into simpler fragments of carbon. And thus we used these two as a key factors to check whether their level were modified with respect to day and with respect to time. From this, you could see like the increase in esterase activity and dihydrogenase activity with the precedence of day until day 5. The maximum of dihydrogenase and esterase were found in bioelectrodegradation compared to the biodegradation. So this could show us an indirect estimation that bioelectrodegradation is better in terms of synergy because the bacteria are stimulated by the presence of electrodes to have a faster, efficient metabolic activity, which in turn results in removal of pollutant at higher rate. Similarly, we want to check the key genes that could show us some idea of why bioelectrodegradation like enzymatic activity, in which we took esterase, PCD, catalase, where PCD is the specific enzyme that is responsible for the removal of phthalic acids, like esters of phthalates. So if you could see like the relative expression compared to the control, the bioelectrodegradations were relatively higher, which in turn supports the synergistic activity of bioelectrochemical systems. So the next, we wanted to optimize the degradation parameters, like as I said, initial pollutant concentration, initial pH, temperature, inoculum dose, and applied potentials. While there are other parameters like external resistance, temperature of shaking incubator, fluctuation of pH by maintaining pH buffer, and light. These are several other parameters that do have potential impacts, but due to the experimental constraints, we were very limited to do with these 5 parameters, in which, if you could see, like at lower concentration up to like 25 milligram or 50 milligram or 100 milligram, there were like almost complete removal at the end of day 8, while there were like a significant reduction in pollutant removal, like 97%, 95%, 94%, 80%, and at the highest concentration of 800 milligram per liter, there were a maximum removal of 75 to 80%. So we do, as based on the MIC data, which we have seen just moments before, the limitation of this also depends on the bacteria's ableness to utilize dibutyl phthalate as a sole carbon source. So there are limitations up to what concentrations which we could work, but that not necessarily limit us to work within that specific concentration. As if we want to go for higher concentration, it's simply that we have to wait for more longer time. Since all the experiments were planned for an 8th day of maximum, so we tend to stop the experiment up to 200 ppm, 250 ppm, 400 ppm, they were much better. Followed by the initial pollutant concentration optimization, we went to check the initial pH as one of the parameters which influences the growth of bacteria. Of all, the neutral pH, which favors the more growth of Bacillus subtilis, therefore pH 7 was also showing the maximum removal rate of dibutyl phthalate in bioelectrochemical systems. And temperature. The Bacillus subtilis tend to have a much stable and optimal growth at 31 degrees Celsius compared to the other temperatures. Therefore, pH and temperature could be solemnly due to the presence of bacterial growth conditions. Therefore, we proceeded further experiments at 31 degrees Celsius, followed by inoculum dose. Since like the inoculum doses were controlled to initiate the system, since it was a closed single batch reactor system, we do not want to top up the bacteria as the reaction slower. So we wanted to check whether there could be any potential impact over the outcome if we increase the initial inoculum dose. But to our surprise, that 1% by volume of overnight grown culture, which was OD was adjusted to 0.1 at 600 nanometer, was well sufficient, and there were no difference even if we increase up to 5% of volume by volume. This could give us an insight that the system which we designed and optimized was much stabler, that even with the minimal amount of bacteria, it was able to stabilize its function in terms of removing dibutyl phthalate with period of time. And finally, the applied potential, which is like one of the heart of bioelectrochemical system, is to optimize the applied potential. Like higher applied potential will increase in applied potential will always favor in removal rate, but not necessarily always. As you could see that the increase in electrodegradation was much higher when the applied potential was increased from 0.5 millivolt to 1 millivolt. Whereas further increase on 1.5 or 2, there were like antagonistic effects. This probably because there were like localization of electron transfer, that bacterial was not able to transfer as an electron shuttle, that bioavailability was hindered. Therefore, higher potential is not necessarily a key factor for higher degradation. So that parameter which we optimized, and from these preliminary experiments, we found that the optimized condition was around like 400 milligram with pollutant concentration and initial pH of 7 at 31 degrees Celsius with 1% and 1 milliwatt, we were able to completely remove the dibutyl phthalate degradation. But this optimization is specific for our system, whereas if we want to scale up the system, we have to do these kind of optimizations specific to the reactor which we are planning to deploy. Further, based on the intermediates which we identified during the process of degradation using HPLC MS, these are the key intermediates which were identified. If you could see, initially the esterase was actually helping in breaking down the complex structures into simpler fragments such as phthalic acid, catechol and phenol, while further degradation of phthalic acid, catechol or phenol and acetaldehyde were favored by the presence of dioxygenase, dihydrogenase, and eventually end up in water and CO2, carbon dioxide, and other gases which were relatively no to nil toxic. But in presence of these intermediates or not necessarily means that almost complete mineralizations were observed. So we want to check the toxicity of the treated effluent based on the intermediates which were detected. So for that, we used toxicity estimation based on ICRISBS using US Environmental Protection Agency software called TEST version 5.1, which gives us a relative idea of what kind of a toxicity these specific compounds do possess. If you could see that initially dibutyl phthalate as a parent compound has a substantial toxicity in developmental and bioaccumulation factor was so bad, while LC50 for Daphnia and fatted minnows were also worrisome. Whereas the intermediates, the toxicity level tend to decline, and eventually the final one of acetaldehyde, which was mild to no to mild toxic, but you could see like there are phthalic acid or phenol or catechol which do possess certain toxic amounts. But we do not have any way of proving that or quantify these specific compounds in the treated effluent, but we just know that we are detected because it varies over time to time. So we wanted to have an estimation of actual experimental method to prove that the treated effluent was toxic free or almost reduced to toxic. So we deployed the next method, which is like evaluating the effluent against peripheral blood mononuclear cells or human PBMC cells in MTTSA. From that, you could see that almost the DI water and DBP, which was treated using BES, had similar or very less toxic compared to the untreated effluent at higher concentration. Even though there is a slight reduction in cell count based on the cell volume based on MTTSA, as per OECD guidelines of 201, it is still within the limits to consider as non-toxic to low toxic. On further of extending this toxicity study, we wanted to check the lethality assay of Artemia, since microplastics do possess a potential toxic impact on marine ecosystems. So we chose Artemia as one of the sample animal model for establishing that toxicity limits estimation in marine ecosystem. So therefore, in which you could see that the distilled water, there were like no to nil damages in the integrity of the organ, while at DBP untreated effluent at higher concentrations, clearly you could see there is a significant rupture in the cells and the structural integrity was compromised at higher concentration, while BES treated effluents was almost significantly similar to that of distilled water. While the graph which you see over here, there is a model representation which says that there is toxic levels for DI water as well. Probably this kind of toxicity could be arised due to the fluctuation in salinity caused by DI water because since it's a marine ecosystem, there is a fluctuation in salinity or pH. There could be slight decrease, so DI water should not have been an ideal candidate to check for this experiment. However, there is a substantial decrease in toxicity compared to the untreated effluent. And finally, we wanted to check its toxicity level. As animal model, we chose mice, in which you could see the body weight of animals up to a course of 10 weeks, which were 10, I'm sorry, 10 days. There was a substantial decrease in body weight for DVP untreated effluent fed animals by oral gavage on every day. 5 ml of oral gavage was fed throughout multiple occasions per day. And from this graph, you could see like there is almost a stable weight was maintained for animals which were fed with DI water and BS treated effluent, while there is a decline in untreated effluent. When we harvested the animal organ post-treatment after 10 days, the animal weight to organ weight was measured. From that, the organ weights of primary organs which we chose like lung, heart, kidney, liver, and spleen. Because in general, these are the organs which do show toxicity in terms of dibutyl phthalate at a very short span. So we chose these 5 as the primary organs. But if you could see that there were like no subtle difference between lung or heart, kidney, liver, and spleen weight for DI water and BES treated effluents were more or less similar, while the animal organ pictures, where these are the representative animal organ pictures, you could see like almost the organs were identical in terms of their integrity compared to control, the DI water and BES treated effluent. While if you notice that there is an inflammation in liver which was seen in BES untreated effluent. So when we wanted to quantify the accumulations, you could see like the DI water which had no accumulation of DBP, while there were similar to less accumulation of dibutyl phthalate which was identified in lung, quantified in lung, heart, liver, kidney, and other organs. But while compared to the BES untreated effluents, in spite they do have accumulated, their levels are much within the limits, therefore no toxicity was observed in the animal which was fed with BES treated effluent. So based on this, we do have a certain idea that BES could be used as an efficient methodology for removal of pollutants, specifically microplastics, in a short time at an efficient rate. So based on this, we could have a summary of what is like. There is a significant synergistic effect by combining biodegradation and electrodegradation. The enzymatic activity and gene expression showed there is a enhanced relative expression in bioelectrodegradation compared to that of biodegradation, and the toxicity of the intermediates which were found, intermediates and the end products which were significantly lower than dibutyl phthalate, and based on the toxicity methods of cell viability was increased while mortality and systemic toxicity were decreased. So with this overall, we do have, we can say successfully that this method can be used in other pollutants as well, but further experimental procedures and deeper understanding on the key enzymes and gene ontology, like going for like advanced techniques such as like single cell sequencing, RNA sequencing, metagenomics, which could help us to understand how this mechanism works within the cell. So I would like to extend my thanks to the National Research Foundation of Korea and Ministry of Education for funding this project. And also I would like to extend my thanks to the Department of Life Science, College of Arts and Science, and the principal and all the department faculty heads for organizing an event to engage with these students. And I feel much appreciated to be in the audience for this one-day conference. Thank you. And if you have like any questions, I would love to have an interaction with these students.
Hello, Dr. Karshavardhan. This is Krishnaveni. Microbiology department. Hope you remember me.
Hello, ma'am. Nice
Yes.
to talk to you. Yeah. Please.
Okay. Is there any consortium effect or consortium of microbes that you tried or your laboratory is working on in the degradation per se?
Actually, ma'am, we do have multiple projects. One of the approach was to first to optimize the system, like a reactor system with a because optimizing a reactor system with a consortia could be challenging because maintaining the consortia at the same bacterial diversity and richness for repeatability applications is quite challenging with our limited resources. So we wanted to optimize the system with a single bacterial system. And since now we have optimized the system for a single bacterial system, it is very much easier for us to control in a consortium environment. So we are actually currently in process of employing consortia, while obviously there is a significant advantage on using a consortia or a mixed bacterial system against a single bacterial system. So yeah, we do have consider and we are in the process of employing that one as well.
Thank you. Because whenever we talk about microbial degradation, of late and the current scenario in the global research and applications tell us strongly that it's not a single organism every time that we'll have to depend on degradation approach. So if any other laboratory is working, so probably with the next visit when you come to India, we would like to learn and understand how that can be adopted and taken up here as part of the research at PSGCAS.
Yeah, sure, ma'am. I would love to actually have an interaction with the students, and I'm happy to share the knowledge which we got from our research team. Obviously, it's much easier for me to share, and I'm happy to share, and I would love to share.
Thank you, Dr. Harshavardhan. Students? Thank you so much for joining us today.
Thank you, ma'am.
Please do visit the college next time when you come to Coimbatore. Looking forward. Thank you. Thank you. Thank you so much. Thank you. And this is a thanks from the student side.
Thank you, sir, for your detailed session about bioelectrodegradation of DPP using LLS-04s from landfills and evaluation of integrated toxicity across various biological models. Thank you once again. Participants, kindly occupy the front seats. Participants, kindly occupy the front seats.
muscle cells that let us smile, the brain cells that perceive the humor in things, the cells of our eyes that take it all in, contain a complete set of all our genes, the genome. If we could journey inside ourselves into a cell, we would see 23 pairs of chromosomes packed into a nucleus. Each chromosome contains a long coil of DNA. If all the chromosomes were unwound, the DNA in just one of our cells would stretch 6 feet long. The DNA double helix contains 4 kinds of building blocks. An A always pairs with a T, a C with a G. DNA contains information to make every part of our bodies with its 4-letter language. Each of our thousands of genes codes for a specific part. RNA polymerase copies the information in a gene into a messenger molecule, messenger RNA. The building blocks of messenger RNA and DNA are called bases. The bases on one strand of the DNA specify the order of bases on the new strand of messenger RNA. The DNA always stays inside the nucleus, but messenger RNA travels out into the cytoplasm. There, a protein-making machine called a ribosome can read messenger RNA to make a particular protein. Every 3 bases of the messenger RNA molecule codes for an amino acid. Proteins are made of amino acids. tRNA molecules help translate the language of DNA and RNA into the language of proteins. tRNA molecules bring the right amino acids that the ribosome links together to make a protein. Proteins are the laborers. Some form structures like tendons and hair. Others perceive light, scents, and flavors, control chemical reactions, and carry messages between cells. To understand our molecular selves, scientists have read the 3 billion letters making up the DNA in the human genome. Different sets of genes interacting with complex environmental factors influence things like our looks, personality, and risks for diseases like cancer and heart disease. A growing understanding of our genes and all they do will help us understand the complexity and the wonder of life. A simple friendship, a sunny day, a life. Studying the human genome, the complete set of human genes, is a way of studying fundamental details about ourselves. A 4-letter alphabet makes up the 3 billion letter-long sequence of DNA that, divided into 23 pairs of chromosomes, inhabits the core of each cell in our bodies. Of course, people are not identical, and DNA sequences do differ subtly between individuals. The Human Genome Project is producing a representative sequence. Separate projects are charting variations in the sequence. The representative sequence is a composite from several people who donated blood samples. Originally, close to 100 people volunteered to give blood and gave their informed consent, affirming that they agreed to the study of their DNA. No names were attached to the blood samples, and ultimately scientists kept only a few. These measures ensured that the DNA sequence would remain anonymous. Not even the donors knew whether their samples were actually used or not. The Human Genome Project aims to read letter by letter the 3 billion units of human DNA. The Human Genome Project scientists began large-scale DNA sequencing in 1999. Before starting to sequence the human genome, they built maps of the human chromosomes and developed and refined techniques for analyzing DNA. With the tools in place, genome scientists took just a year to amass sequence covering more than 80% of the genome. The human genome is a massive text. If the 3 billion letters, called bases, that make up all the human DNA in a cell were printed in telephone books, the stack containing the whole genome would reach as high as the Washington Monument. To figure out the sequence of all the bases in the genome accurately, scientists need to read the 3 billion bases not just once, but at least 6 to 10 times. Sequencing reactions can only reveal the order of a few hundred letters of DNA at a time, amounting to a fraction of a page. Having many overlapping segments of sequence allows the genome to be puzzled back together into an intact whole. Before shredding the DNA in the genome and starting to sequence, Human Genome Project researchers first built a map of the genome. They found thousands of landmarks scattered throughout the chromosomes to help them navigate among all the DNA. Developing genome maps was a key step to prepare for DNA sequencing, but the increasingly detailed maps have also been an important tool orienting hundreds of geneticists hunting for disease genes. With enough markers in place, the Human Genome Project scientists created libraries of clones that span the genome. Each of the clones contains a manageably small fragment of human DNA that is stored in bacteria. Scientists can tell what part of the human genome each clone derives from by figuring out what markers each contains. This clone-by-clone approach for analyzing the human genome makes it possible to double-check the locations of sequence. And because the Human Genome Project has been an international effort with many laboratories taking part, carving up the genome has allowed different groups to coordinate their work effectively. Clone libraries offer the advantage of real libraries, orderly access to information. In most clone libraries, fragments of human DNA are stored in a kind of bacterium, E. coli, that normally lives in our large intestines. Each E. coli cell in a library stores a single segment of human DNA, so that the human fragment can be tracked and copied easily. To sequence the human genome, Human Genome Project scientists cut relatively large clones called BACs, which are 100,000 to 200,000 bases long, into smaller fragments. The smaller fragments, which are about 2,000 bases long, are typically stored in E. coli viruses. The scientists determined the precise order of the larger clones because pinpointing the positions of many smaller clones is much more work. But for actual sequencing reactions, the smaller clones are more suitable. E. coli cells containing fragments of human DNA can be stored in freezers indefinitely. When researchers are ready to retrieve DNA from the library, they revive the cells by bringing them back up to 37 degrees centigrade, gut temperature. To make many copies of the human DNA, the E. coli cells act as copiers. A few containing the same bit of human DNA inside them are so the
Learning is the product of active engagement and participation. As we commence the final session, it is my privilege to invite Dr. J. Senthil Kumar, Assistant Professor, Department of Biotechnology, PSGCAS, to introduce our distinguished chief guest, Dr. N. Senthil Kumar, Scientist F and Bioprospecting Division, ICFRE, Institute of Forest Genetics and Tree Breeding, Coimbatore, Tamil Nadu. The dais is yours, sir.
Good afternoon to one and all present here. With a pleasure, on behalf of the Department of Life Science, I'm happy to welcome Dr. Senthil Kumar N., who is currently working as an a research scientist in IFGTP, Coimbatore. And I'm very much thankful for accepting our invitation, sir. I welcome you, sir. And to little about Dr. Senthil Kumar, his area of research is entirely about bioprospecting of forest resources for the benefit of the humankind. And sir has been working, his academic qualification is BSc Zoology in specialization with entomology in 1994, MSc Zoology in entomology in 1996, and MPhil in Zoology specialization with entomology in 1997, PhD Zoology with specialization of entomology in 2003. And coming to sir's work experience, from the year 1997 to 1999, he worked as ad hoc lecturer in Madras College at Madurai. And sir has also completed his research and carried out as a researcher in 2003 to 2008 at a Rainforest Research Institute, Jharkhand, Assam. And during the year of 2008 to 2009, sir has served as a scientist in Forest Research Center, Hyderabad, Andhra Pradesh. And from the year 2009 to till date, sir is working as a scientist in Institute of Forest Genetics and Tree Breeding, Coimbatore. To add feather on his cap, sir has worth in mentioning, sir has secured about around 7.2 crores of projects, research projects, inclusive of 25 research projects, which is funded by ICFRE, BIRAC DBT, ACRC DST, and ACRB DST, NRDMS DST, and Ministry of Environment and Forest, NBA, are the few agencies to be mentioned here. And apart from getting grant from the specialized agencies, sir has also discovered 5 species, out of which 4 species are from Arthroptera species, and they have been published and also deposited in Zoo Bank. Sir has also produced various products and technology. Till date, 12 products has been transferred, and 12 eco-friendly products like biopesticides will become high act, tree pal, entophyte nasa, crawl, clean, Simca BC, and biobooster, Tara red natural colorant jam, and Royal Sima Rand Sander soap, anti-arthritic spray, herbal hand sanitizers, are the few other. And apart from this, sir has also a honorable recipient of 3 patents and a trademark and copyright for 5 products, which is already available in the market as Tree Rich Booster, IFGTP Herbal Hand Sanitizer, Tree Pal, Red Sander Soap. And sir is also having copyright for few other mobile applications and few other industry-related, tree-related projects. And to noteworthy, he is also holding 110 research papers in leading research journals, both international and national, with 15 books and written about 18 chapters. And he has also carried out around 45 training programs on bioprospecting, which to benefit the student community, teachers, and the officers of IFS, scientists, academics, and research scholars. So Sir is also being a reviewer for project proposals in DST, DBT, and he is also being a reviewer in various other journals also. And to that, he is also, to wind up with my speech, in short, if you want to put a single word for Sendil sir, a man of bioprospecting. Other words, you can Google it and you can find out a lot more information. It's just to give a brief introduction about Dr. Sendil. And with that few words, I just thank the committee and Sendil sir for accepting our invitation to being served as an resource person. Thank you.
Thank you very much, Dr. Sendil, Dr. Prabhakaran, and other dear colleagues. I am being in touch with Dr. Sendil right from 2010, it seems. Yes. So one of his students have come for a short-term project, and since then we are in touch. And then Dr. Prabhakaran, we had a blue grant scheme with Bharatiyar University. I was the coordinator for forestry sector. The advisor, Dr. Shankaran, is keeping in touch with us still, but he is always telling that you have very good projects, number of projects that has to see the funding agencies. So we have a close association with PSG College of Arts and Science. At the outset, I would like to thank the principal of this college and faculty members, Dr. Sendil and Dr. Prabhakaran, and other faculty members for this opportunity. I think you are all from the northern side. I can talk in English as well as in Tamil also, no? No problem at all. Okay, thank you very much. So, as sir has introduced, நான் எழுதி கொடுத்துருக்கேன், அதனால தான் அவ்வளோ படிச்சிருக்காரு. இன்னும் நிறைய எழுதி கொடுத்து இன்னும் நல்லா சொல்லியிருப்பாருன்னு நினைக்கிறேன். But anyhow, thank you very much. I don't know whether worth for it or not. Kindly pardon with me, I am not good at lecturing. So way back I worked as 1997 as a lecturer in Majra College. But I am not good at lecturing, that is why I left the job and then I have gone into research. Kindly pardon with me. So just I would like to share my research experience with you all. This is a very big crowd, but unlike teachers, we scientists, if we have the politicians mic kadacha vidamaatanga, so students kadacha scientists vidamaatanga. If you visit any research institutions, if you meet any scientists, they will never leave you because alla arnu aruthu eduthiruvanga. So don't think that if you feel bored and just raise your hands, there is no problem at all. I am not your teacher, your principal, not your supervisor. You can go out and be back. Okay. So with this small what's called request, and then I would like to thank our organizers So they are all my teachers. Professors always a teacher, whether it's a scientist or is a president of India. So teacher is always a teacher. We respect teachers. So I respect all the teachers over here and the scholars. So let us start my business. Work on one more. Okay, thank you. Sorry. Yeah, this is the institute where I am coming from. It is Institute of Forest Genetics and Tree Breeding. It is one of the regional institutes of Indian Council of Forestry Research and Education under Ministry of Environment, Forest and Climate Change. Our core competence is genetics and tree breeding. You might have seen this building in one of the films. Nanban padam pathingla. So this is our headquarters. It is Indian Council of Forestry Research and Education at Dehradun, Uttarakhand. This is our headquarters. Our core competence is genetics and tree breeding. So we tailor-made trees for our what you can say for our stakeholders. It is paper industry, pulp industry, plywood industry, and then matchwood industry, pencil wood industry, and so on. So because what happened, and we are importing lots and lots of wood from other countries, even from Thailand, Burma, for our domestic needs. So we tailor-made trees for various stakeholders based on the genetic improvement and then biotechnological interventions along with the silvicultural operations and all. This is what we are working on different stakeholders. So with this small introduction where I am coming from, we need to have a different outlook. So definitely we need to have a different outlook. What is that outlook? So you might have think that without these things, can we imagine our life? Can you say yes or no? இது ரெண்டு இல்லாதவங்க யாரா இருக்கீங்களா? Anybody in this auditorium? Anybody in this forum? Can we imagine our life without these things? இது ரெண்டு இல்லாதவங்க யாரா இருக்கீங்களா? Okay. Cell and genes are very, very important. Let me come to later on. Okay. So Abraham Waxman, Ukrainian-born US scientist, he has invented what is called streptomycin for tuberculosis. Cordyceps sinensis. This, yeah, it is entomopathogenic fungus which is available only in the eastern sub-Himalayas. It is infested by, the fungus is infested on these particular lepidopteran insects. It is available in particular region of eastern sub-Himalayas and particular season only, but it is having high value material because it is used in hyperglycemia, renal dysfunction and then anti-tumor activity. Manganine, you see the Xenophis is a South African frog. The manganine is a pigment used for dog bites. Leeches, so Hirudinaria, you might be knowing that. See, it produces a material called hiruidine. It is equivalent to heparin. You know very well that what is bald headedness. You know the men are suffering from this bald headedness than the women because it is an autoimmune disease. So if you go to vascular surgeon, they have to have hair implantation. So otherwise, if you go to any what's called skin doctor, they used to tell that you have to apply coconut oil and you have to rub it vigorously. The hair, this what you can say, the blood will be what's called circulated more and more, and then hair follicles will be retrieved. But this, if you have the Hirudinaria, these leeches, and it oozes out the blood, it sucks the blood, and it aggravates the blood circulation. Automatically, hair follicles will be retrieved. So that is why one of my friends, when I had a DST training in the year 2009, is one of the Ayurvedic doctors. He asked me to supply this what's called leeches. Now I worked in Northeast India. The leeches in Northeast India is totally different from the Southern India because there it is very long one. We call it as Rama and Krishna leeches. They used to tell that it's very long one. But here in Valparai, if you go to this area, tea estates, you will get these leech bites, you will get fever immediately. Still I have the leech marks with me. Even sometimes I used to have the mite attack every year. So I have gone to a skin doctor, he told that you just apply coconut oil and it will be there for another 1 year. Meanwhile, I will go again to the forest area, and especially this teak forest area. If you get into that one, you will get the mite attack very easily. So be careful with that one. Okay. So if we have leeches, we need not to go to a vascular surgeon for the hair implantation, because what automatically hair will be retrieved due to aggravation of blood circulations. The Zevia, zero calorie sugar plant, it is being used in the beverages, because with the diabetics, India is the #1 country as far as population as well as diabetic and pre-diabetic is concerned, because of the fatty liver. So that is one of the important area in which we have to do some research. So we need to have zero calorie sugars for beverages. And and all the plants, almost all the plants are having the chemical cues. We like smell, isn't it? We use perfumes, isn't it? Otherwise the people cannot sit aside, because what will happen, the sweat smells, because it is, we call it as amino acidemia. So what will happen, we human beings, insects also like smell. If it is like smell, it is a lure. If dislike a smell, it is a repellent, like for example mosquito repellent and all, we can say. So all the plant material, they have the chemical cues which may be used as a lure or a repellent. And what not from insects, since I am an entomologist, and then honey, silk, and then varnishes, everything is from insects. So என்னடா அவன் பாட்டுக்கு slides போட்டுக்கிட்டே இருக்கான். What is the link? What is the commonality? There is a commonality among the slides that we have seen. So we have the commonality is, and minds of products are with us in the natural resources. It is our duty to tap those resources, to make it into a product, is called bioprospecting. Colloquially telling, கோழி கடைக்கு போனா உயிருடன் கோழி ₹100 இல்லையா? If it is dressed one, it is 200 rupees to 230. If you go to KFC, it is 400 rupees, 500 rupees. This is nothing but value addition to the resources. This is called bioprospecting. Bioprospecting is not a new thing. It is a very old term, because we develop products based on our natural resources. It's called bioprospecting. India is one of the biodiversity rich countries in the world. Everybody talks about the diversity of India. We have 8% of the world's species, biodiversity countries, 2 lakhs plants, 90,000 animals, almost around 16,000 sacred groves, almost around 700 protected areas. As far as bioprospecting is concerned, we have only 2%, but China is about 15%. China is far ahead with us. So we have to beat the China. We have beaten the China with our population, but we can't beat with the China for technological interventions. And since India is one of the biodiversity rich countries in the world, and we are the biopirated country in the world. What is biopiracy? Biopiracy is nothing but நமக்கே தெரியாம நம்ம பாக்கெட்ல இருந்து காசு எடுத்து ப்ராடக்ட் வாங்கி திரும்ப நம்மகிட்ட வித்து காசு வாங்கிடுது. This is called biopiracy. The best examples are neem, basmati, and then turmeric. Thanks to CSIR, we got back all the patents back, because it is available in our traditional system of medicine. So what is the solution for what is the solution for this biopiracy? The only solution is bioprospecting. Our Prime Minister used to say that there is Atmanirbhar Bharat. So we have all the resources in our hand, but we need to develop some products and we can make money out of it. This is called bioprospecting. I would like to tell you some of my what's called experiences and how to go about bioprospecting. I told you that we have 2 lakhs plants and 90,000 animals and microorganisms. How to search the product, how to search the queue. So குப்பையில ஊசி எத்தற மாதிரி அலைஞ்சிட்டு இருக்க முடியாது. So we need to follow some criteria. The first one is indigenous knowledge. We have our forest dwellers, indigenous people. They have the huge knowledge of their own traditional system of medicine. So we have to acquire their knowledge and then we can go for identify the compounds from the plants or microbes or whatever it may be. Then availability criteria. For example, if it is neem, tulsi, nochi, it is available in plenty. So, we can use it sustainably for production of various products for the ailments. And biodiversity criteria. For example, if it is a Sirocco asoka, Taxus baccata, they are very, very, very rare and endangered. We can identify the compounds. For example, if it is Cinchona, quinine can be isolated, it can be synthesized separately. So, likewise, we can synthesize the compounds which is very rare and endangered from the plant material. So, these are the 3 different criteria we do follow for bioprospecting of forest resources. The first story I would like to tell that in 2009 and 10 everybody was scared to fly from India to other country to India, and this is because of the swine flu. The remedy for swine flu is Tamiflu. The precursor for production of Tamiflu is cichimic acid. This is what the student of Dr. Sendil is collaborated with me. So, the cichimic acid is available plenty in star anise, the Chinese star anise. What happened? The China started or stopped to export this Chinese star anise to other countries at that time, because everybody should depend on China for the star anise to extract cichimic acid. All the countries, they are looking for cichimic acid for the production of the Tamiflu. Another 2 steps from cichimic acid we can get Tamiflu. So, we are also working on this production of cichimic acid, and we found that this is the girl, and we found that this Iconia grossipus is agaya. So, we have come across what called a paper that this the weed can be controlled by a weedicide called glyphosate. How the glyphosate works on it, because it stops the glycolysis cycle, the scrub cycle in the cichimic acid itself. So, what happened, and we apply this glyphosate on these aerial parts of this what you can say, this agaya thamarai. We stopped the cichimic acid pathway in the cichimic acid itself, and we have harvested more and more quantities of cichimic acid. This is called the this work that has been conducted by this Lenora. Sendil sir is also there. And we have also identified an endophyte which can able to produce the same cichimic acid more and more quantity. So, you see the Chinese star anise only 0.63%, but water hyacinth will give 3.25%. Another 2 steps we can go for production of Tamiflu. And you might have seen the rotten lemon and oranges at your home, but something is left over by this fungus, but it can't able to engulf all the lemon. So, something is there, this something is nothing but statin. Akira Endo is a Japanese biochemist who has come out with the statin material. India is #1 country for statin. Statin is good for, it is only drug for cholesterol control. You believe it or not, I am taking 40 mg of atorvastatin every day. I am 52 years old. என் ஒய்ஃப் காலையில் எழுந்திரிச்சு 5:30 எழுந்திரிச்சு மருதமலை பக்கத்தில் இருக்க வீடு, பாரதியார் யூனிவர்சிட்டி பக்கத்தில் 2 கிலோமீட்டர்ஸ் ஓடணும், 2 கிலோமீட்டர்ஸ் நடந்து வரணும். காலையில் ஒரு 2 சத்து மாவு உருண்டை கொடுப்பாங்க, மத்தியானம் 2 சப்பாத்தி கொடுத்து விடுவாங்க, நைட் 2 தோசை கொடுப்பாங்க. அவ்வளோதான், நோ நான் வெஜிடேரியன். Even though I am taking 40 mg of atorvastatin because of my family history, strong family history. I told you that there is a fatty liver. So, though we have taken more and more quantities of carbohydrate, it will be converted into fat material, it will be deposited in the adipose tissue. That is why we have to reduce the fatty liver in order to avoid the diabetics as well as the cholesterol. Okay. So, we have get into some other research activities. So, this is what the statistics says, that India is #1 country for statin market. It is almost around 300 crores now, it increased to 3 to 4 fold. And we identify an endophytic fungus which can able to produce the alternate to statin, it is cystosterol, that we have identified through GCMS analysis. And another one story is that, and I worked in Northeast India. There are different ethnic groups are there. They used to take insects as a food and medicine. So, we are working on this allantoin is a blowfly component. It is a peptide. It is an antiseptic peptide which has been isolated from these insects. And these ethnic groups use ants for hepatitis B, what's called treatment, cockroaches for asthma and tuberculosis, locusts for tetanus. So these are the that documentation of traditional knowledge you might be knowing that that we have acquired from these ethnic groups from Northeast India. I would like to just tell you that one day I have gone into Mizoram market, the last end of our India, just border of Burma. There is a Mizoram market, and they kept silkworms and the limnaea, you know, the snail, this pond snail. So one gentleman, he has come and then he picked up on this silkworm, tasted it. It was a live insect, live larvae, he tasted it. I was little bit shocked, and then I asked that what for you are tasting it, and I would like to purchase few larvae. He would like to make curry out of it because it is having full of protein, sericin and fibroin. Likewise, and another one gentleman, he has taken this snail and he has tasted it. So I asked that what you will do with this one. He told me that it is used for making soups out of this one. So the people are generally taking medicine, insects and other zoological material or organisms for food and medicine. இன்னும் ரொம்ப கேவலமானதெல்லாம் இருக்குது, சொன்னால் நீங்கள் உங்களுக்கு வாமிட் வந்துடும். ஸோ நாகாலாந்தில் வந்து நாய்க்கறி கேள்விப்பட்டிருப்பீங்க. You believe it or not, I have seen there are shops with dogs. There is a very peculiar delicious dish. என்ன செய்வாங்க, நாய்க்கு நிறையா சாப்பாடு கொடுப்பாங்க, பயிர் நிறையா சாப்பிட்டுட்டு அந்த நாயை எடுத்து சுற்றுவாங்க. சுற்றினோன்னு கிருகிருன்னு வரும் இல்லையா? ஸோ அது வாமிட் பண்ணும். இந்த வாமிட் பண்ணதை எடுத்து டிஷ் பண்ணி சாப்பிடுவாங்க. திஸ் இஸ் வாட் தி Naga peoples are doing. So we have seen obviously. So there are different ethnic groups, they have their different setup of their food behaviors. So even some of the countries, even if you go to Uganda, there is a senene, the grasshoppers are being eaten. They just remove the what's called wings of grasshoppers, and then they can wash it with water and put small quantities of salt, and then can fry it, and they can take it with liquor. It is available only in the winter season. We call it, they used to call it as a senene during the winter season. Even cake toppings are with insect parts. The days are not far away. We need to have the lab, what's called cultured meat for our, in order to meet. Because என்னன்னு கேட்டிங்கன்னா, நீங்கள் மட்டன் ஷாப்புக்கு நீங்கள் போகிறீங்களா என்னன்னு தெரியல. Even though I am a vegetarian, turned vegetarian, for my children we go to purchase meat from market, no. So I have gone there and I asked that where from these goats are coming from. He told that it is from Maharashtra. Nothing else from southern India, because it is very, the population is very, what's called declining by day, because it is one of the promising field. We need to have the cattle breeding in southern parts of our country, because we are in dearth of these meats and all. So I think this one, it's going up, no. Off and turn. Okay. Yeah, our population is increasing in an exponential rate, like we call it as population explosion. So Norman Borlaug from Texas A&M University, along with Professor M.S. Swaminathan, so he told that biotechnology is the only remedy for the global food demand. I think, yeah, of course. Sorry, I am not familiar with this. Yeah, okay. So next slide. Yeah, this one. So this is Norman Borlaug used to tell that this biotechnology is the only remedy for what you can say in order to meet the global food demand. So again, I come to this slide once again. Can we alter our gene and cells as we like? Isn't it? Are you agree with me? என்ன கலர் வேணாலும் மாற்றிக்கலாம், என்ன வேணாலும் செஞ்சுக்கலாம். So can we alter our original cell and genes? Say yes or no? Yes, that is genetic engineering. So we can cut and paste This is called genetic engineering, nothing but biotechnology, as you are all studying. Okay? Yeah, next one. Thanks to biotechnology, we have to slaughter almost around 3 lakh crores. 3 lakh crores of pigs to be slaughtered in order to meet the insulin demand of the globe. Thanks to hybridoma technology, in a matter of seconds, we can produce quantum of insulin for our diabetics. The stenophylla plant, after 32,000 years, the Siberian squirrel swallowed the seeds of the Siberia, this what you can stenophylla, and frozen under the ice. After 32,000 years, they have been retrieved from the stomach of the Siberian squirrel, and they have gone into tissue culture technique. Finally, they got it. This is called species recovery program. So, thanks to biotechnology. Yeah. I think it's worth continuing, or shall I stop? Okay, let me continue. Okay, for another 5 minutes. Don't worry. So, our Green India mission, I am come to my what's called hardcore business. Our Ministry of Environment and Forests used to tell that we have Green India mission 2030, and we need to cover 33% of our land mass should be covered with green cover. This is called forest cover. We don't have land, but we have to depend on farmers for green cover. This is what Green India mission says that. So, obviously, we have to go for barren land covered with green covers or tree biomass, and then all these things. There is no doubt at all, unlike our what you can say, like our agriculture field, if we go for any monoculture, because farmers, they will not simply what's called go for tree cultivation in their farmlands, because they have to have some money, they have to fetch some money, otherwise they will not depend on tree crops for their farmlands. So, we have to go for large scale plantations of Casuarina, Eucalyptus, Melia dubia, Melena arborea, these kind of short rotation, high yielding tree varieties, otherwise the farmers will not adopt it. So, if we go for monoculture of anything, any crops, there will be an insect infestation and then disease prevalence. So, chemical insecticides are panacea. You know very well that endosulfan is banned in what's called in our it's called Kerala. Because of these deleterious effects, we need to go for alternate pesticides, is called biopesticides. This is what we are doing it in our Institute of Forest Genetics and Tree Breeding. So, we have what's called come out with a product called High Act. We have come out with Wilway, High Act, Tree Pod and all, and the High Act is Hypnocorpus pendandra based seed oil based biopesticides. As I told you that this seed oil is having very peculiar compound is called hypnocorpic acid. The hypnocorpic acid, I told you that we use perfumes, isn't it? Because the people cannot sit aside because it is aminoacidemia in our sweat. So, during our master's degree, and we have one program called chronobiology, the mosquito biting cycle. Dryer போட்டுட்டு ஷார்ட்ஸ் போட்டுட்டு ஒரு டார்க் ரூமில் உட்காந்து போனாங்க. ஸோ என்ன ஆகுன்னு கேட்டால் ஈவினிங் வந்து கொசு எவ்வளோ கொசு வந்து கடிக்குது, எங்கே கடிக்குது, எவ்வளோ நேரம் கடிக்குது, எத்தனை கொசு கடிக்குது, ஸோ அப்படின்னு கவுண்ட் பண்ணிக்கிறணும். வி கால் இட் அஸ் இஸ் அ மஸ்கிட்டோ பைட்டிங் சைக்கிள், இஸ் குரோனோபயாலஜி. யாரெல்லாம் ஓ பாசிட்டிவ் கை தூக்குங்க. அவங்களுக்குலாம் நிறைய கொசு கடிக்குன்னு சொல்லுவாங்க. பார்த்து அது தி ரீசன் ஐ டோன்ட் நோ எக்ஸாக்ட்லி, பட் இஃப் யூ சி நிறைய தலையில் கொசு வரும், தட் இஸ் டிஃப்ரெண்ட் ஆஸ்பெக்ட். பட் இப்போ உங்கள் பக்கத்தில் ஃப்ரெண்ட்ஸ்லாம் இருக்குன்னா ஓ பாசிட்டிவ் பீப்புள் ஆர் மோர் சசெப்டபிள் டு mosquito bites. So, what will happen? The mosquito used to come and bite us. What will happen? The mosquito recognize our body smell through their odorant biting protein in the odorant biting site in the sensilla of the antenna. You know why? It is very, very important that the smell of individuals are unique, like our fingerprints. The smell of you is totally different from another girl. The smell of me is totally different from another one. So, the smell is unique like our fingerprints. So, mosquito will come and recognize the smell of the host and come and bites us. And what will happen? It recognize the smell by this odorant binding site. Likewise, this hypnocorpic acid is a peculiar cyclopentenyl fatty acid. It has the peculiarity to bind with important binding protein of the mosquito sensilla. So what will happen? The mosquito will not recognize its host, it is disrupted. So what will happen? It will not recognize the host. Likewise, we also use this echinocorpic acid for recognizing the host plants by the various insects. Microbial based biopesticides. They are host specific, they are very specific to the organisms, but neem is the only oil based biopesticide for insect control, and Karanj oil, the pungam oil, is for the disease control. The rest we can't find it out, because oil based biopesticides are having the sustainability in the market, long term sustainability. We developed this product, the shelf life is almost around 5 to 6 years. That is why it faces the market compatibility as well as the competition. That is why we are always looking for oil based, it is non edible tree borne oil based seed oil for developing a biopesticides. So we have given as a biopesticide to Tanjore farmers, and one of the farmers he used for what's called his paddy field, but we developed for our forestry crops. So he called me in the 2 days later and he told that, what you have given, sir? One of my friends in our adjacent field, he used urea, but he didn't have that lush green growth, but your product is giving lush green growth, what it is there. So we thought that it is only a biopesticide, but we get back to the research with the feedback from the farmers. We found that there are two different compounds, that is caryophyllene oxide and aromodendron oxide, which are responsible for biostimulant activity of the crop. So that is what we filed a trademark and we got it for the what's called tree pall. So I told you that we do work on non-edible tree-borne oil seeds for developing biopesticide, because it has the long sustainability in the market. And another one story, we are working on energy sector. Food and energy sector is very important. We need fossil fuels and more, because you know very well that right from 30 rupees, 50 rupees, 70 rupees, 90 rupees, now it is more than 100 rupees petrol and diesel prices. We have a demand, but the exploration of oil, you know very well Digbai is the #1 first oil refinery in Northeast India. So we have seen all these things, though we have a huge resources in Northeast India for fossil fuels, still we have a demand of fossil fuels. There is no doubt at all, in future we need to go with like this only, otherwise of late we have come out with what we can say, these electric vehicles and all have come, otherwise we need to go like this only, even though because vehicles are coming up in an enormous rate. So that is what we are working on, biodiesel production. We have identified a peculiar plant, the seed oil based biodiesel, and we identified it is a very good biodiesel. We are going to patent it. That is what I not disclose the research part of it. So we would like to, and we are working on colors. Everybody like colors, isn't it, boys and girls, isn't it? We do like colors, though we are studying in the boys college or boys school. So everybody looks colors, isn't it? Looking for, but organic colors. Everybody is looking for organic colors. So we have colors around us, isn't it? So we have flowers, we have fruits, we have lots and lots of colors, even from the soil, even from the insects, even from the what you can say plant material. We can extract colors. Since I am from forestry sector, I am looking for colors from forestry. Oh. I am not familiar. You can operate it. Okay, thank you. So I am from forestry sector. We are looking for colorants from tamarind. It is a red tamarind. Red tamarind is one of the mutant variety. It is not a normal tamarind. It is a mutant variety. Again, this SARS student worked with this tamarind, sir, Lenora. What happened? And this next one, please. So what happened? The colorant, it is 1 in 1000. இந்த செம்புலின்னு சொல்கிறாங்க, இல்லாட்டி ரத்தப்புலின்னு சொல்கிறாங்க. இது எங்கே இருக்குன்னு கேட்டிங்கன்னா சதர்ன் பார்ட்ஸ் ஆஃப் அவர் தமிழ்நாடு அண்ட் தென் நார்தன் பார்ட்ஸ் ஆஃப் வெள்ளூர் அந்த ஏரியாவில் இருக்குது. மோஸ்ட்லி இட் இஸ் அவைலபிள் ஒன்லி இன் த கிரேவ்யார்ட்ஸ் இன்வேரியபிளி, வெதர் இட் இஸ் அ மாஸ்க் ஆர் Hindu religious graveyards or Christian graveyards. It is only available in the graveyards. ஒரு தடவை என்ன ஆச்சுன்னா தேனி டு கம்பம் ரோடு ஹைவேஸ் பக்கத்தில் ஒரு சுடுகாட்டில் ஒரு 2 மரம் இருக்குது. இது நாங்கள் உள்ளே இறங்கி அந்த மரத்தில் காய் பறிச்சுட்ருக்கோம். நானும் இன்னொரு பையனும். ரோட்டில் போகிறவங்க எல்லாம் நிற்கிறாங்க, அப்படியே நின்று எங்களை பார்த்துட்ருக்காங்க. நாங்கள் என்னமோ சரி நம்ம ஏதோ தப்பு பண்ணுறோம் போட்டுருக்கு, அவங்களுக்கு தெரியாமல் அங்கே இந்த இடத்துக்கு வந்துட்டோமோன்ட்டு நாங்களும் இப்படி பார்த்துட்ருக்கோம். அவங்க நின்று இப்படி பார்த்துட்ருக்காங்க. பார்த்தா அங்கே நாங்கள் சுடுகாட்டில் நின்றுட்டு இருக்கோம். சார் சுடுகாட்டில் என்னடா பண்ணுறீங்க நீங்கள் அப்படின்னு கேட்டாங்க. சுடுகாட்டில் தான் அந்த மரம் இருக்குது அப்படின்னு சொல்லிட்டு அந்த மரத்தை 2 மரத்தையும் வெட்டிட்டாங்க அவங்க. ஸோ பயந்துக்கிறாங்க, ரத்தப்புலின்னு சொல்லிட்டு அவங்க என்ன செய்றாங்க, எல்லோரும் இன்வேரியபிளி மாஸ்க்லையும் வெட்டிட்டாங்க, வெள்ளூரில் இருக்கக்கூடிய மாஸ்கில் வெட்டிட்டாங்க, கிறிஸ்டியன் சர்ச்சில் வெட்டிட்டாங்க. ஸோ we have identified 32 accessions of this red tamarind and almost 50% of the tree have gone by this one way or the other. So luckily we have a germplasm bank with us. So we have extracted cyanidin 3-glucoside. It is pH dependent and then temperature dependent, light dependent compound that we have identified. It is responsible for red in color. We filed a process patent, we got it. So this one, and we tried for textile dyeing, but we could not come out because any natural dyes which are not good for textile dyeing. This is what we got experience. But finished product we can like shirt பண்ணிட்டு அதை பண்ணி we can use it. But for color fastness, even though we can use it with some chemicals for modern, so color fastness is not as much a good one. So even this, it gives very baby pink color. But colorant for textile industry is depending on the demand from other countries. During summer they look for baby pink color, during winter they go for golden brown color kind of thing. So we thought that why don't we, since it's a tamarind, why don't we go for food colorants? We found that it is very much what's called good with jam preparations. We prepared a jam, and then so while taking jam, this Lenora, she found that the colorant is stick onto the lips. She told that, sir, it is very good, but it is having very good whitening property. Though it is retained for a long time and it can be wiped out very easily. So we thought that why don't we go for preparing a lipstick. We have come out with a lipstick from red tamarind, even red sanders and Caria arborea. That also we filed a patent. So we got a FSSI, and then this technology, this is what I got 2 DST projects that will transfer this technology to the tribal welfare. We are transferring technologies. Next one, please. So we work on medicinal aspects of this wilvam. You know wilvam? Wilvam fruit is good for stomach, you know very well. Oh, you are here only. Okay, thank you. Sorry to disturb you. Okay. So wilvam, we are working on wilvam, and then know very well that our teacher யாராவது திட்டி விட்டாருன்னு வச்சுங்க, பசங்களை கேர்ள்ஸ் முன்னாடியோ கேர்ள்ஸ் பசங்க முன்னாடி திட்டி விட்டா தூக்கம் வராது இல்லையா? திட்டி விட்டாரு அப்படி இப்போலாம் கவலைப்படுறதில்லை இல்லையா? அப்படி தான் என் ஒய்ஃப் ஒரு நாள் என்னை திட்டி விட்டா. என்ன பண்ணா, இவ்வளோ பெரிய சயின்டிஸ்டாக ஆச்சான்னு ஒரு நாள் திட்டி விட்டான். எனக்கு தூக்கமே வரல. என்னென்னமோலாம் சொல்லி பார்க்குறா, கன்வின்ஸ் பண்ணி பார்க்குறா, சரின்னு தோணுது, ஆனால் தூக்கம் வரல. காலையில் 8 மணிக்கு புரூக் ஃபீல்ட்ஸில் போய் உக்காந்துட்டேன். புரூக் ஃபீல்ட்ஸ் திறக்கலை, ஆனால் வெளியில் உட்காந்து என்னடா செய்கிறதுன்னு சொல்லிட்டு ரொம்ப இதாகிட்டு இருக்கும்போது என்னோட கைட் சூப்பர்வைசர், ஐ டூ ரிமெம்பர் எவ்ரி டே பிகாஸ் அவர் ஃபோட்டோ முன்னாடியே இருக்கும், டாக்டர் KP சஞ்சயன். rarely he used to call me. He called me on that day. I was, hi Arun. Okay, thank you. Good morning. Thank you. ஓ நிறைய பேர் இருக்கீங்க. ஓகே தேங்க்யூ. ஸோ திட்டி விட்டாங்க. அவர் கூப்பிட்டாரு. கூப்பிட்டு என்ன பண்ணுற அப்படின்னு கேட்டார். சார் இந்த மாதிரி என் ஒய்ஃப் திட்டி விட்டா சார் அப்படின்னு. அவர் சொன்னார், நீ ஃபாரஸ்ட்டில் ஒர்க் பண்ணுற, நீ வாட்டுக்கு ஊராக சுற்றுற, காட்டுக்குள்ளே போகிற, நல்லா அனிமல்ஸ் பார்க்குற, நல்ல பிளான்ட்ஸ்லாம் பார்க்குற, ரிவரில் குளிக்கிற, ஃபால்ஸில் குளிக்கிற, உன் ஒய்ஃப் என்ன செய்வா, வீட்டில் தானே இருப்பா, அவள் திட்டாமல் யார் திட்டுவா அப்படின்னு கேட்டார். ஸோ ஐ ரியலைஸ்ட் இட். ஆனால் எல்லாம் புரியுது சார், ஆனால் தூக்கம் வர மாட்டேங்குது, என்ன செய்கிறது. ஸோ திங் இஸ் தேட், ஸோ ஹி டோல்ட் மீ, யூ கோ அண்ட் மீட் அ டாக்டர் and then you will get rid of from this one. I met a doctor, he has given only one pill, and then I got sleep and I slept. The thing is that, so our hypothalamus used to secrete a hormone called serotonin. Serotonin is a balancing hormone, okay? It balances the stress and strain. இங்கே உங்களே அறியாமல் நீங்கள் தூங்கும்போது நீங்கள் கேள்வி பார்த்துருப்பீங்க, திடீர்னு டக்குன்னு ஒரு சுவிட்ச் ஆஃப் ஆகும். எப்போ ஆகுதுன்னே உங்களுக்கு தெரியாது. இந்த சுவிட்ச் ஆஃப் ஆகுது பார்த்தீங்களா தூங்குறதுக்கு, இட் இஸ் பிகாஸ் ஆஃப் த செரோட்டோனின் கண்டென்ட். பட் அவர் ஹைப்போதலாமஸ் யூஸ் டு செக்ரீட் செரோட்டோனின் டு பேலன்ஸ் அவர் ஸ்ட்ரெஸ் அண்ட் ஸ்ட்ரெயின், பட் த ப்ரிகர்சார் ஃபார் செக்ரீஷன் ஆஃப் தி செரோட்டோனின் ஷுட் பி டேக்கன் ஃப்ரம் அவுட் சைட். நீங்கள் உங்கள் பாட்டி தாத்தாலாம் சொல்லியிருப்பாங்க, நீங்கள் தூங்கும்போது லைட்டாக ஒரு lukewarm milk குடிச்சிட்டு தூங்கணும்னு சொல்லுவாங்க. This mainly because of the milk contains the precursor, the moiety to aggravate the hypothalamus to secrete serotonin. We found that this wilwam fruit is having that kind of moiety to aggravate the serotonin content. Next one please. We have a tie-up with the PSG College of Pharmacology, Dr. Ramanathan sir. So the animal was put it under stress, then they have administered this drug and the animal was able to swim and then face the light. This kind of animal test we have conducted. So we identified the serotonin compound which are able to produced by the hypothalamus of this animal Wister rat by administering this wilwam fruit. பெரிய ஹைட் அண்ட் சீக் ட்ரக்லாம் ஒன்றும் கிடையாது. ஸோ வில்வம் ஃப்ரூட் எடுத்து நல்லா தண்ணி விட்டு அரைச்சி சுகர் போட்டு குடிச்சிட்டா போதும், வேறு ஒன்றும் கிடையாது. ஸோ அதவே எடுத்து என்ன பண்ணுவோம், சுகர் போட்டு லயோஃபிலைஸரோ இல்லை ஸ்ப்ரே ட்ரையரில் போட்டோம்னா பவுடர் ஆயிரும். பவுடர் தூக்கி கேப்சூலில் போட்டோம்னா திஸ் இஸ் கால்ட் another one please. This is called R500, nothing but we have the scientific proof, scientific validation for the stress and strain. Next one please. Red Sanders, புஷ்பா படம் பார்த்துருப்பீங்க இல்லையா? புஷ்பா படம் பார்த்தீங்களா? The red sanders is this red sander. It is available only in the particular area. It is endemic to the Rayalaseema area of Andhra Pradesh. நீங்கள் திருப்பதி போயிருந்தீங்கன்னா பார்க்கலாம். திருப்பதி மேலே போகும்போது பூரா இருக்கக்கூடிய ரெண்டு சைடு இருக்கக்கூடியது பூரா அந்த ட்ரீ தான். நீங்கள் ராஜம்பேட், சித்தூர், கடப்பா அந்த பக்கம்லாம் போனீங்கன்னா நீங்கள் பார்த்தீங்கன்னா யூ கேன் see the huge plantations of the red sanders. ஒரு வாட்ச் டவரில் ஏறி பார்த்தீங்கன்னா எங்கே பார்த்தாலும் ரெட் சாண்டர்ஸாக இருக்கும். Everywhere you can see red sanders, not even a single other species. The red sanders is having very peculiar kind of density. It is having very iron rich material that is having the very high value product, wood product. இப்போ நம்ம திருவண்ணாமலைக்காரங்களாம் பயங்கர கெட்டிக்கிறாங்க. There are different grades of red sanders. இப்போ பிளைனாக இருக்கும், ப்ரௌனிஷாக இருக்கும், ரெட்டாக இருக்கும், வேவி கிரைன் இருக்கும். The wavy grain is very very important, very high value wood. அதை கண்டுபிடிக்க தெரிஞ்ச ஒரே ஆட்கள் யாருன்னு திருவண்ணாமலையில் இருக்கக்கூடிய ட்ரைபல்ஸ் தான். இப்போ கேள்விப்பட்டிருப்பீங்க, சுட்டு கொண்டுட்டாங்க இல்லையா, ஒரு 22 பேர் 23 பேர். லைவ் ட்ரீயிலே வந்து வேவி கிரைனை கண்டுபிடிக்கக்கூடிய கெட்டிக்காரத்தனவங்க ஜவாதி ஹில்ஸில் இருக்கக்கூடிய ட்ரைபல்ஸ் மட்டும்தான், வேறு யாராலையும் கண்டுபிடிக்க முடியாது. அதை அவங்க என்ன செய்வாங்க, அதை வெட்டி டீபார்க்கிங் பண்ணி பார்க்கை எரிச்சிட்டு மரத்தை மேலேருந்து கீழே இறக்கிடுறாங்க. மலையிலேருந்து கீழே இறக்கிடுவாங்க. ஒரு ஆள் தூக்கிட்டு வர முடியாது இந்த சைஸ் உள்ள மரத்தை. ஐ திங்க் அருண் நீங்கள் வந்திருக்கீங்களா? வரல, நம்ம NDF ஸ்டடீஸ். ஸோ ஒரு மரத்தை ஒருத்தர் தூக்க முடியாது, 2 பேர் தான் தூக்க முடியும், மினிமம் 2 பேர். மலையிலேருந்து கீழே இறக்கிட்டா ஒரு டன் 1 லட்ச ரூபா. ஆந்திராவிலேருந்து தமிழ்நாடு பார்டர் வந்துச்சுன்னா 10 லட்ச ரூபா. தமிழ்நாட்டுக்குள்ளேருந்து சென்னை பார்டர்லேருந்து சென்னைக்கு வந்துடுச்சுன்னா 20 லட்ச ரூபா. போர்ட்டுக்கு போயிடுச்சுன்னா 30 லட்ச ரூபா. சைனா போயிடுச்சுன்னா 50 லட்ச ரூபா. ஜப்பான் போயிடுச்சுன்னா 60 லட்ச ரூபா. This is the wood value of these red sanders. It is available plenty. So we thought that why don't we work on bioprospecting aspect. Wood நீங்க எடுத்துங்க, bark நான் எடுத்துக்கிறேன், நீங்க எதுக்கு எரிக்கிறீங்க. So bark we have worked on this one, and then we have come out with a cosmetic products called the soap, ICFRE Royal Sema. It is not a Royal Sema, it is a Royal Sema. So we got a trademark. Why I have put this figure, பார்த்தீங்கன்னா picture, எல்லாரும் நினைச்சிருக்கூட ரெட் சாண்டர்ஸ் போட்டால் நம்ம நல்லா ஃபேர் ஆயிடுவோம், ஃபேர் அண்ட் லவ்லி மாதிரி. இட் இஸ் நாட் லைக் தட். பட் glow skin. Instead of the fair skin, you will get a glow skin because it is having this very what's called good medicinal properties. It heals anti, what you can say, it's not only a moisturizing content, it is having this skincare properties. That is why we have come out with the soap and a cream from red sanders. Next one please. Yeah, we have also come out with lipstick. It is a very good lipstick, it is available with me, and we filed a patent but we have not commercialized it. This is the red sanders Kerry arborea. Another one bark we used it, that we have made it for hair dye also, Kerry arborea. Next one please. We do work on anti-cancer properties from this papaya for malaria, this one what you can say, dengue fever, and then this ICRISBS for cyclamic acid, even for Annona muricata. One of the girls from the same Sri College, she came out with Annona muricata for anti-cancer properties, and then Pterocarpus indicus is good for another anti-cancer properties. Still the things are there to come out with the products. Next one please. Yeah, so I am taking much of time, isn't it? I will finish it within 5 minutes. Okay, no problem. No problem. Shall I finish it or? Okay, okay. So Cinchona we worked on during first COVID wave, and then Trump, you might be heard about that. Trump insisted our India to what's called supply hydroxychloroquine. So we are the colonial, during our colonial period, and we found that the quinine is good for anti-malarial purposes. The Cinchona from the Peru, it was named after her, and then so we have large plantations of Cinchona. The Cinchona bark is having 31 alkalides, but we had a GCMS analysis, we found almost 131 alkalides. Next one please. And so quinine is not water soluble, that is why we always take it with the salt form of quinine, that is hydroxychloroquine. So what happened, one of my friends during school days, he is a schoolmate, he is orthopedic doctor. நம்ம நிறைய வாட்ஸ்அப் குரூப்லாம் வச்சுருக்கோம் இல்லையா. ஸோ ஸ்கூல், பிரைமரி ஸ்கூல், ஹையர் செகண்டரி ஸ்கூல், யூஜி, பிஜி, எம்ஃபில் நிறைய குரூப்ஸ் இருக்குது. அதில் one of my friends, he is an orthopedic doctor, he told that so he used to prescribe hydroxychloroquine for arthritis problem, but it has some side effects. So he told that, Senthil, why don't you come out with some topical applications. So we have Volini, no? Volini is the only one, is a what you can say diclofenac based painkiller, is pain spray you can say. So we thought that why don't we go for developing a pain spray with Cinchona bark extract. During our period we used to have turpentine, you might be heard about turpentine. ஏதாவது அடிபட்டுச்சுன்னா எங்கள் அம்மாலாம் வந்து கொடுப்பாங்க, டர்பன்டைன் தேய்ச்சி விடுவாங்க. ஸோ இட்ஸ் அ பைன் ஆயில். ஸோ வி புட் டுகெதர் பைன் ஆயில் அண்ட் தென் Cinchona bark extract to come out with a pain spray called Walk Easy, that we filed a patent and we got it. Next one. So and the second COVID wave, and then I was asked to go for developing a product. You know very well that there is the COVID, SARS COVID-19, the virus used to attack in the upper respiratory tract. You might be heard that the சின்ன குழந்தைகளுக்குலாம் அது அட்டாக் ஆகாதுன்னு சொல்லி கேள்விப்பட்டிருப்பீங்க. ஏன்னு தெரியல உங்களுக்கு. ஏன்னு கேட்டிங்கன்னா there is a binding site, angiotensin converting enzyme binding site. அது வந்து சின்ன குழந்தைகளுக்கு very primitive. So only for we adults we have the highly developed these binding sites. That is what the spike protein will come and bind it on it. So for that one, this angiotensin converting enzyme should be activated. It aggravates our binding site to accept the spike protein. So what will happen, we need to stop the ACTU enzyme to accept our receptors to bind the spike protein. So we have some of the compounds which has been isolated from the different plant material, we made it into a inhaler, because if the compounds will go inside and it mimics the angiotensin converting enzyme binding site, so it will not accept the spike protein of the virus. So we found that it is being there, but it is not, we have not come out with this one, this is in evaluation stage, not only for SARS-CoV, but any kind of viruses that can be, which is responsible, even for SARS and then MERS virus we can use it for this one. Still we have come out with even Herbal's hand sanitizer in the first COVID wave we have come out. Next one please. Sanitizer நான் தயார் பண்ணா யார் நம்புவா, நம்ப மாட்டாங்க இல்லையா, யார் வேணாலும் சொல்லிக்கலாம் hand sanitizer ன்னு சொல்லிட்டு. But we have given to the BioLine laboratory whether it is really having the sanitizing property or not. So they have this system of evaluation. So before application the 10 வரலையும் culture plate ல வெச்சிருவாங்க. After application every 5 minutes interval they have to keep it on culture plates, they have to see what is the culture of organisms. So it has been drastically reduced from 100% to 3%. You have seen the table here. So really they said that is really a herbal hand sanitizer, but we have to follow the WHO norms like 75% to 85% of alcohol should be there. Next one please. We are working on this CRISPR based technology for gene editing technology for developing some of the ailments. Our biotechnology department is doing for some of the what's called stress tolerant or environmental tolerant crops. Next one. It will be used for various diseases, that will be very good. Even for PPO in apple, browning of apple can be reduced by what's called restricting the PPO enzyme by the CRISPR-Cas based technology. Next one. Next one please, go ahead, go ahead. So what is the need of the hour? The very easiest way of doing MSc dissertation work is to get any plant, go for extraction, go for our HPLC GCMS analyze pannite, we can publish a paper. There are about 2 lakhs papers every year come out based on identification of the compounds, but there is no translational phase. It has to face the industrial partner, it has to come out as a product. So what will be the what's called input? If you take turmeric and pepper alone, the efficacy will be different. But rasama kudichamna, the efficacy will be much more. It is 20,000 times fold. Next one please. So what will be the solution? We need to have tie up with industry. For example, we have lots and lots of patents. So NBA, we have to get a clearance from NBA for filing a patent. The Raghuram, he used to tell that Dr. Sendil, and we are getting enormous applications, even patent of Dr. Thanga Pandian, he used to tell me that. So we have received lots and lots of patents, applications, most of them are promotional patents. So they will not face any what's called industrial applications or commercial aspect. I have got all these 3 patents because I have a tie up with industry, then we filed a patent and got it. So we need to have all the interventions or innovations should face the market, otherwise it will be like this one only. Okay. So our study should not be like this, and we need to have different outlook, as I told you in the first beginning of my presentation. So what will be the strategy? We have resources, forest area, the resource owners are forest dwellers. We R&D people can do characterization and then tie up with pharmacological colleges, go for animal testing, tie up with clinical trials, tie up with the industry to come out with a product, and automatically our economy will grow. This is called bioprospecting. Next one please. So we need to have the current science. Excellent. Okay. So we need to have the current science instead of the static science. What is the, you know very well that our curriculum should align with the what you can say current science. It should what's called align with the what can say, the commercial aspect of the products. As I told you that this is small quantities of the research that we have done for last 20 years, 2 decades. So, Senthil Kumar has come out with 12 and odd products. You are all hundreds of people, maybe you can come out with hundreds and hundreds of products, because we have 2 lakhs plants and 90,000 animals. Still there are number of plants and animals yet to be described. They need to have the commercial aspect. Next one please. So, there is no doubt at all, technology is boon or ban, it is in our hands. So, thank you friends, thank you very much for your patience listening. So, due to time constraints, I have restricted my what's called presentation, and then lots and lots of things are still there with me. I forgot to tell you that because I thought that there will be a different forum, because you are all students. We have huge opportunities in forestry sector. I will tell my story, and then I don't know Hindi. During 2002, 2003 I posted in Assam first time. I have gone there, and then my first posting, 5 years I spent there, and after 3 years I had a due for my promotion, right from scientist B to scientist C. We started with scientist B, C, D, E, F, G. So, I have gone to my assessment every time. We have to go for our headquarters for our promotion, and then we have all the materials with me to hold another position at the higher level. My Director General, he simply told, Senthil, you are looking so young, you go there and be back on another year. He did not assess me, and he told that, sir, I want transfer rather than my promotion. He got angry, and then he said, you will be neither given promotion nor transfer, you go to Jorhat, Assam and be back on next year, I will give you. At that time, one of my expertise, he came out, and then he told that Dr. Kujar from IARI, he told that Senthil, he was my external examiner, he told that why are you sticking on to this sector? You shift to ICIR, you will be flourished. But I told him that, sir, this is my passion, and then this is close to my heart, and I like this forestry sector in my career. So, I did not left this position, though I got another one opportunity in DRDO. So, I stick on to the forestry sector, because forest will give you lot many things. Even our scholars, even Arun knows that girl also knows that. If anybody comes to IFGTB, they will not leave IFGTB, because we have huge opportunities, and then huge opportunity to visit different forest areas. So, the thing is that we have JRFs, we have technical assistant posts. Recently, we have conducted a examination for technical assistants, technicians, scientists, IFS officers. All these things are open to you all. It will open a new door for you to get into the forestry sector. நீங்கள் ஏதோ ஒரு கஷ்டமாக இருக்குது, நீங்கள் ஒரு பார்க்கில் போயிட்டு திரும்ப வந்தீங்கன்னா you will be refreshed. So, இது என்னோட எக்ஸ்பீரியன்ஸ்னு சொல்ல வரல, but you all love natures. So, that is what I told you, that you keep watch our website, Institute of Forest Genetics and Tree Breeding. Huge opportunities are there. We are in dearth of good students for our research projects, and we are in need of your support to come out with these kind of products. So, this is not my output, these are all my students' output. Tomorrow you will be our one of our scholars, and then tomorrow you will come out with some product. Again I will put your picture, and I will give some lectures in some other college that these are all my scholars' output. So, this is what I request you. It is not my just information, but I request you all to get into forestry sector, at least agriculture sector, So you will be flourished. With this one, I would like to tell you only one thing. நீங்கள் இப்போ எல்லோரும் சின்ன குழந்தைங்களா இருக்கீங்க, உங்கள்கிட்ட சொல்கிறதுக்கு வந்து எனக்கு ரொம்ப இந்த இவ்வளோ தூரத்துக்கு சொல்கிறதுக்கு வந்து காரணம் என்னென்னு கேட்டிங்கன்னா, தோ யூ ஆர் ஆஃப்டர்நூன் ரொம்ப நல்லா சாப்பிட்டு தூக்கம் வர மாதிரி இருக்குது, எனக்குமே தூக்கம் வந்துச்சு. இருந்தாலும் நான் சொல்கிறத காது கொடுத்து கேட்குறீங்கன்னா, there is only one thing, because you know during Second World War the நாஜி படைகள் எல்லாம் வந்து ரஷ்யாவோட லெனின் நகரை ஆக்குப்பை பண்ணிட்டாங்க. So there are about 2.5 years the continuous war, and during 1894 I think so. So Nicholas Vavilov, you might be heard about that, is a tree, not a tree, there is seed center in Leningrad in Russia. 1920 ல இருந்து 1930 ல 10 years almost around 65 countries ல இருந்து 1 lakh seeds collect பண்ணி அங்கே வச்சிருக்காங்க. During Second World War ல என்னன்னு கேட்டிங்கன்னா, Leningrad during the winter period, the Leningrad நகரத்தை டோட்டலாக வந்து இந்த நாஜி படைகள், ஹிட்லரோட நாஜி படைகள் வந்து ஆக்குபை பண்ணிட்டாங்க. The huge food demand, யாருக்கும் சாப்பாடு இல்லை, குளிர், எல்லாத்தையும் சாப்பிட்றாங்க. நாய், பூனை, எலி எதை கிடைச்சாலும் பிடிச்சி சாப்பிட்றாங்க. மரப்பட்டைகளை உரித்து காய வச்சு குடிக்கிறாங்க, பெல்ட்டு வச்சு காய வச்சு குடிக்கிறாங்க. ஒரே ஒரு ரொட்டி துண்டுக்காக தன்னை உடம்பே விற்கிற அளவுக்கு ஒரு தாய் ரெடியாக இருக்கா. அறிவாளியான குழந்தை இருக்கட்டும், மிச்ச குழந்தைங்கள்லாம் சாகட்டும்னு கொண்டு போடுறாங்க. இவ்வளவு சூழ்நிலையிலையும் கூட அந்த சீட் சென்டர்லேருந்து ஒரு சீடை கூட அவங்க கொள்ளையடிக்கலை. ஏன் தெரியுங்களா? இந்த சீடுங்கிறது இந்த நாட்டினோட சொத்து. The seed is the treasure house of the nation. ஒரு சீடை கூட அவ்வளோ பசியிலையும் பஞ்சத்திலையும் பட்டினிலையும் ஒரு சீடை கூட அவங்க என்ன செய்யலை, கொள்ளையடிக்கலை. அவ்வளவு உலகத்துக்கே சோறு போட்ட நிக்கோலஸ் வாவிலோவ், ஃபாதர் ஆஃப் சீட் சயின்ஸ் யூ கேன் சே, இந்த உலகத்துக்கே சோறு போட்ட நிக்கோலஸ் வாவிலோவ் பட்டினியால் செத்து போனான் அப்படிங்கிறதான் நிதர்சனமான உண்மை. நான் ஏன் உங்கள்கிட்ட சொல்கிறீங்கன்னா, டுடே யூ ஆர் த சீட். இன்றைய வித்துக்கள், நாளைய விருட்சங்கள். நீங்கள் தான் இன்றைக்கி நான் போட்டிருக்கக்கூடிய சீட், டுமாரோ வில் பி தி விருட்சம். ஏன் அதை சொல்கிறேன்னு கேட்டிங்கன்னா, தோ வி ஆர் ஒர்க்கிங் ஆன் என்னென்னலாம் சொல்கிறோம், பயோடெக்னாலஜி அது இதுன்னு சொல்கிறோம், பட் விதையில்லா விருட்சமும் விந்தில்லா மானுடமும் அழிந்து போகுங்கிறதா உண்மையான நிதர்சனம். ஸோ வித் திஸ் ஒன் ஐ வுட் லைக் டு சே தட் தேங்க்யூ வெரி மச் ஃபார் யுவர் பேஷன்ஸ் லேர்னிங். ஒன்றே ஒன்று மட்டும் ஃபைனலாக சொல்லிடுறேன், பேசிகிட்டே இருக்கானு நினைக்க வேண்டாம். 10 வயதில் அறியாமையோடு இரு, தவறில்லை. 20 வயதில் அழகாக இரு, 30 வயதில் அறிவோடு இரு, 40 வயதில் செல்வத்தோடு இரு, 50 வயதில் அமைதியாக இரு, 60 வயதில் ஆற்றலோடு இரு, 70 வயதில் சந்ததியோடு இரு, 80 வயதில் சந்நிதியோடு இரு, 90 வயது வரை இருந்தால் நிம்மதியாக இரு. நன்றி, வாழ்த்துக்கள். தேங்க்யூ.
Thank you, sir, for your informative session regarding innovations in bioprospecting sustainable utilization of forest resources. Now I request Dr. J. Sendilkumar, sir, to honor our resource person. If anybody have queries about the last session, you can ask the doubts now. If anybody have doubts about the last session, you can ask now. Valediction is about recognizing the contributions and achievements of others. Let us acknowledge and celebrate the ideas exchanged and the milestones achieved throughout this conference. It is my immense pleasure to invite Dr. N. Krishnaveni, Assistant Professor and Head, Department of Microbiology, PSGCAS, to deliver the welcome address.
We have gathered here at the end of a day-long discussion on various aspects of biological sciences. Good evening, one and all. I take this opportunity to welcome our guest, Dr. N. Senthilkumar, Scientist F, Chemistry and Bioprocessing Division from the Indian Council of Forest Research and Education, IFGTB, Coimbatore. I welcome our beloved principal, Dr. Sengutuvan sir, the organizer, Dr. Victor Arogyadas, Associate Professor and Head Department of Biochemistry, Dr. Uttara Madam, who's going to present the report of the one-day program, Dr. Thamare Chelvi from Food Processing Department. I welcome all the heads of various departments, faculty members and staff, and my dear students for this valediction of one-day conference that we have gathered here. We had a lot of deliberations from morning, from gut microbiome to bioprospecting and various applications, and took lot of initiatives today on how research approach, particularly the audience here are postgraduate students, sir, who are listening to the session. So I guess every student would have taken up the insights on how their learnings can be applied, as Dr. Senthilkumar highlighted, how our learnings can be taken to applications and to the benefit to the society rather than putting it aside as publications. So with this insight, we are very happy that we could take up with such a short notice a very fruitful seminar for the whole day. And I welcome one and all for this valediction function. Thank you.
Thank you, ma'am. Now, I would like to call upon Dr. L. Uttara, Associate Professor and Head, Department of Nutrition Dietaries, PSGCAS, to give a report on today's conference.
With pleasure, presenting the report of the conference, Recent Innovations in Sustainability and Biological Sciences. Dr. Victor Arogyadas, Associate Professor and Head Department of Biochemistry, welcomed the gathering and stated that platforms like these have immense scope to widen and expand scientific explorations. A preamble about the conference was presented by Dr. Senthilkumar, Associate Professor and Head Department of Biotechnology. In his presidential address, Dr. M. Sengutuvan, Principal in Charge, emphasized the need for connectivity, convergence, and exchange of information for growth of research in a direction that will benefit the society at large. He also added that scientific innovations should not disrupt ecosystem to ensure sustainable holistic growth. Dr. Kumar, Scientist G, and Center Head DRDO DIBT stressed that evolution of technology should augment human performance realistically and should transform the lives of people with a positive impact. As the world is facing resource crisis, research and technology should focus on providing real-time solutions for bettering the lives of human beings. He further expressed DRDO's readiness for development of basic research in academic institutions as well as opportunities for students to associate with DRDO. Vote of thanks was proposed by Dr. S. Dhanakumar, Assistant Professor, Head Department of Environmental Science. The resource person for technical session 1, Dr. Kumar, in his session on gut microbe and postbiotics, unlocking their potential for human health, was in alignment with the famous quote, "Death begins in the colon," that originated with Nobel Prize-winning Russian biologist Eli Metchnikoff, who postulated toxic products of intestinal bacteria are responsible for diseases cited as early as 1960 and has become frontier research today. Dr. Kumar highlighted on factors influencing gut microbiota and protective role of postbiotics. Metabolic products and bioactive components generated by lactobacillus bifidobacteria have positive health implications. In improving microbiota profile, colored fruits and vegetables provide tremendous benefit to gut microbe and also reduce reactive oxygen species. As military persons are exposed to high levels of stress, probability for imbalance in gut is high, and research in favor of symbiotic association in gut that will modulate health has been prioritized in DRDO, he further added on. The next session on integrated recirculation of aquaponics, a sustainable aqua agriculture system, was taken up by Dr. P. Santhanam, Professor Marine Planktology, Department of Marine Science, Bharathidasan University, Tiruchirappalli. He emphasized aquaponics as a viable tool to solve food and water crisis and ensure food security. He dealt with the components, types of aquaponics, and symbiotic relationship between microflora, waterbed, and plants that ensured higher productivity, phytochemical, and nutritional components. As the system is devoid of chemical fertilizers and pesticides, sustainability is ensured due to zero waste productivity. Dr. Harshavardhan Mohan, Postdoctoral Associate, Division of Biotechnology, Jeonbuk National University, South Korea, highlighted the importance of bioelectrodegradation of dibutyl phthalate using landfill isolated Bacillus subtilis. Using animal models, he has studied the deleterious effects of environmental toxins on vital organs. The session on innovation in bioprospecting, sustainable utilization of forest resources by Dr. N. Sendil Kumar, Scientist F, Chemistry and Bioprospecting Division, Institute of Forest Genetics and Tree Breeding, Coimbatore, expressed bioprospecting using indigenous knowledge and scientific research has promising potential both in promoting health as well as in therapeutic conditions. The economic and medicinal importance of insects was also expounded. Sir has explored into seed-based oil pesticides, biocolorants, and alternative biofuel sources and biocosmetics. He also emphasized translational research requires industry-academia collaboration. About 451 participants took part in the conference. Thank you.
Thank you, ma'am. We are so happy to have active participants in ICRISBS 2025. It's time to hear from our participants about their learnings on today's conference. Kindly share your valuable thoughts and feedbacks with us. Thank you.
So good evening. I'm not a microbiology student. I'm a faculty representing Karunya Institute of Technology and Sciences, respected dignitaries on the dais and off the dais, and student friends. I'm here to share the importance of the conference on sustainability and biological sciences. So this conference has been a platform. I'm a faculty. I'm teaching undergraduate students as well as post-graduation students. And I know what is the difficulty which actually the students community are facing. Even after having an education, what next is an important question. For some, it should be a placement. For some, it should be an higher studies, either towards post-graduation or towards research. So I trust and believe that this conference has provided every one of us the idea starting from material science, and especially in the last session, nearly around 15 to 20 products which has been developed from the different resources which are available in our own environment, especially from the forest resource alone, has been shared with the students community. So it was, I thank you, sir, for enlightening us towards product development, which is the need of the hour. So every research, it has to reach to a level of application, and all the speakers of this conference has emphasized on the recent concepts and the technology, what each one of us has to understand, and how far we can make use of it in our own research towards product development. And the other important aspect as student community, what an awareness has to be created among the student community is developing products and moving on towards commercialization, that is entrepreneurship. So this conference has given the platform for all the students as well as the faculty members like me from other institutions to gain confidence that we can also move towards patents and products development. So I take this opportunity to thank the management of PSG and all the authorities of PSG Arts and Science College and the heads for providing us an opportunity to be part of this conference. Thank you so much.
Good evening. So, இந்த ஒன் டே கான்ஃபரன்ஸில் வந்துட்டு எல்லா லைஃப் சயின்ஸ் ஸ்டூடெண்ட்ஸும் வந்து இருப்பீங்க. ஸோ எல்லாருக்குமே வந்து இந்த 4 செக்ஷனுமே வந்து ஒர்த்துன்னு கேட்டிங்கன்னா அது ஒர்த்துன்னு தான் நான் சொல்லுவேன். ஏன் அப்படின்னா ஃபஸ்ட்டு வந்துட்டு மெயினாக வந்துட்டு கட் மைக்ரோபயோம் அப்படிங்கிற டாப்பிக்கில் ஃபஸ்ட்டு எல்லாருமே பேசினாங்க. எல்லாருக்குமே வந்து கட் மைக்ரோபயோங்கிறது எல்லா லைஃப் சயின்ஸ் ஸ்டூடெண்ட்ஸ்க்கும் தெரிஞ்சிருக்கும். ஸோ காமனாக தெரிய வேண்டிய அந்த லைஃப் சயின்ஸ் ஸ்டூடெண்ட்ஸ்க்கு கட் மைக்ரோபயம் பற்றி தெரியும். ஆனால் அதோட ரிசர்ச் பற்றி அதிகமாக நம்ம யாருமே அவ்வளோவா டெவலப் பண்ணியிருக்க மாட்டோம். ஜஸ்ட் ஒரு ஐடியா கிடைக்கிறதா இருக்கட்டும். அது மட்டும் இல்லாமல் எல்லா செக்ஷனுமே ஒவ்வொரு செக்ஷன்லேயுமே வந்து ஒவ்வொரு வேல்யூபிள் பாயிண்ட்ஸ் அண்ட் வேல்யூபிள் ஐடியாஸ் வந்து நம்ம எல்லாத்துக்குமே கிடச்சிது. ஸோ இந்த கான்ஃபரன்ஸ் வந்து பர்சனலி எனக்கு வந்துட்டு 100% ஒர்த்துன்னு சொல்லுவேன். ஸோ தேங்க்யூ ஃபார் த டிபார்ட்மெண்ட் ஆஃப் லைஃப் சயின்ஸ் அண்ட் ஹூ ஆர் ஆர்கனைசிங் திஸ் ப்ரோக்ராம் வெல் சார். தேங்க்யூ.
Good evening everyone, myself Jayashree from Department of Biochemistry. Today's session was very useful. We have learnt about the different aspects of the life science. First session which was completely belongs to the gut microbiome, and which was very useful how our body works with the help of gut microbiome. And second session also very nice about the aquaponics, which deals with how fishes are grown in different types of aquaponics, and we have gained lots of ideas about the business on the concept of aquaponics. And third session about the biodegradation is also very useful to us. And then the last session, thank you for the last session, sir. We have gained lots of knowledge about the last session, about the trees and different products you have made, and different products, their knowledge. Thank you, sir. Thank you one and all present here. Thank you.
Thank you, participants. With great respect, I invite Dr. N. Sendil Kumar, Scientist F and Bioprospecting Division, ICFRE Institute of Forest Genetics and Tree Breeding, Coimbatore, Tamil Nadu, to present the valedictory address. We look forward for your concluding words for today's gathering.
The most respected principal of this college, distinguished guests on the dais and off the dais, esteemed faculty members, colleagues and friends, and dear student friends. Very good evening to all. I feel very honored and privileged that I have been invited to conclude this important event on recent innovations in sustainability and biological sciences. You all are witnessed for insightful discussions and groundbreaking research and a visionary perspective that has the power to transform sustainability in forest sector, or most importantly biological sector, today and tomorrow. You have explored many innovations that will help us to think about sustainability in health sector, not only in health sector and biological sectors. The convergence of various disciplines in life sciences is paving a way for newer solutions of sustainable health and biological sciences and today and tomorrow. There is no doubt at all, dear children, that innovations are cornerstone of progress. But this important event might have demonstrated you to face today's challenge of the world. Recent technologies like CRISPR-based gene editing technology is opening a new door for not only treating various ailments, but also preventing what's called various diseases in an effective manner. And also you have discussed about, I think, the morning session as well as in the afternoon. You might have discussed about the interdisciplinary approach of various disciplines in the biological sciences. But integration of traditional knowledge with modern science is need of the hour, because we need to have meet the challenges of the today's world with respect to climate change, newer diseases, and then health disparity and biological diversity. And also I would like to extend my gratitude to the organizers for well organization of this conference. And most importantly, the innovations with respect to sustainability is no longer be the theoretical concept. It is also a practical imperative. We need to have innovations with recent advancements in health sector and biological sciences, which should be affordable to the general public and accessible to the general public without compromising the environment of the health or health of the environment. And we need to have the sustainability in conservation of biodiversity as well as utilization of biodiversity in a sustainable manner. For that, we need to have more and more innovations, which is in the hands of you young minds. With this one, I extend my gratitude to the organizers and all the participants who have contributed your research experience and then your time, passion, and knowledge, which is truly inspiring me. And today we have laid a foundation stone for tomorrow's healthier society. I appreciate and congratulate the organizers. You brought all the young minds and then beautiful, brilliant minds in a single platform, which is truly inspiring, which is truly a memorable event. And I would like to urge you all to commemorate this event, to innovate, to cooperate and coordinate and advocate a healthier society tomorrow. With this one, and thank you all. Thank you very much for your continued endeavor. Thank you very much. Thanks for permission. Thank you.
Thank you, sir. Now it's time to award the winners of the today's presentation. Now I cordially invite Dr. K. Lavanya, Assistant Professor, Department of Biochemistry, PSGCAS, to announce the winners. And also, I request the resource person to distribute the certificate to the winners. Thank you.
Oral presentation. First prize goes to Kavinila S., Research Scholar, Biotechnology, Karunya Institute of Technology and Science, Coimbatore. Second prize goes to Pooja Krishna, GS, Second MSc Applied Microbiology, Department of Microbiology, PSGCAS. Third prize goes to Aishwarya G. Nambiar, Research Scholar, Department of Biochemistry, PSGCAS. For poster presentation, first prize goes to VM Nandini, Research Scholar, Zoology, PSGCAS. Second prize goes to Akshaya M., Department of Biotechnology, Karunya Institute of Technology and Sciences. Third prize goes to Dayalin CKM, Department of Biotechnology, PSGCAS.
Gratitude is the best attitude. I now request Dr. M. Thamare Selvi, Associate Professor and Head UG, Department of Food Processing and Technology, PSGCAS, to propose the vote of thanks.
Gratitude is a powerful catalyst for happiness. It is my pleasure to propose vote of thanks to all the dignitaries who have contributed to the success. I place my sincere gratitude to our Managing Trustee, Sri L. Gopalakrishnan, for granting permission to conduct this international conference. I take this opportunity to record my thanks to our respected Secretary, Dr. T. Kannayan sir, for constant support and guidance in all our activities. I express my deep gratitude to our inspiring Principal in Charge, Dr. M. Sengatuvan sir, who has been the driving force behind all our academic endeavors. Thank you, sir. I extend my sincere thanks to our Vice Principals, Dr. M. Jayanti and Dr. M. Umarani, for their moral support and guidance. I extend my profuse and deepest gratitude to our esteemed Chief Guest, Dr. R. Kumar, Scientist G, Center Head, DRDO DIBT, DFRL Mysuru, for accepting our invitation, for inaugurating the seminar and deliver the keynote address on gut microbiome and postbiotics. At this juncture, I would like to thank Dr. Kadirvelu sir for being instrumental to invite our chief guest for this conference. Our warmest thanks to our eminent speaker, Dr. P. Sandhanam, Professor, Marine Planktology and Aquaculture Laboratory, Department of Marine Science, Bharathidasan University, Trichurapalli, for his insightful speech on aquaponics, a sustainable agriculture production system. Thank you, sir. I sincerely thank our resource person, Dr. Harshavardhan Mohan, Postdoctoral Research and Associate, Division of Biotechnology, John Buck National University, who joined us through online from South Korea. I wish to express my wholehearted thanks to distinguished resource person, Dr. N. Sindhil Kumar, Scientist F, Chemistry and Bioprospect Division, Institute of Forest Genetics and Tree Breeding, Coimbatore, for gracing this occasion and delivering the valedictory address. Thank you, sir. I would like to thank our principal, Dr. D. Brinda ma'am, for initiating the collaboration and strengthening the unity of life science departments and promoting interdisciplinary synergy in organizing the conference annually. I would like to take this opportunity to thank our Dr. Victor Arogyadas sir for meticulous and dedicated planning and execution of this conference successfully. I extend my sincere gratitude to the heads of all the life science departments for their relentless support to conduct this conference. I would definitely thank our faculties of life science departments and especially all the organizing committee members who work tirelessly by dedicating their time and effort to make this event a grand success. I express my sincere thanks to the participants from various institutes like Karunya and SSM College, etc. A special note of appreciation to our oral and poster presenters for their active and enthusiastic participation in presenting their research work. I sincerely acknowledge the unwavering support rendered by the research scholars and student volunteers for their relentless efforts behind the scene, ensuring smooth execution of the program. I extend my heartfelt thanks to our technicians and non-teaching staffs for their support and cooperation for the event. I must mention a special thanks to the Department of Visual Communication and ITMC for the video and audio coverage and live streaming in YouTube channel for the online participants. I thank the Maintenance Department for the uninterrupted technical support leading to the smooth conduct of this program. I thank the food court for supplying us with a delicious lunch and refreshment. I place my gratitude to the press members present here for capturing and disseminating the highlights of the conference in print media. I should definitely place my gratitude to all the students participants from various disciplines of our college, unless whom the conference should not be this grand success. Sustainability is not just a topic for discussion, it is a responsibility we all share. As we part ways, let us carry forward this spirit of sustainability and innovation, working together to build a greener, healthier, and sustainable world. Thank you all once again, and we look forward to seeing you at future conference. Thank you.
Thank you, ma'am, for your warm words. Now let us stand for National Anthem. refreshments for the participants is arranged in front of the auditorium.